Hello and welcome to Taconic's webinar with the exciting title, A Mouse is Not a Mouse is Not a Mouse, Genetic Background Matters, which will be presented today by Dr. Gerald Bothe, who is a senior scientist in Taconic's R&D department. My name is Margit Wissenbach and I will be your moderator today. Before we get started, I'd like to address a few practical matters. This webinar will be made available to all attendees. We will email a link for the presentation to you as soon as possible. Directly after this presentation, Dr. Bothe will answer your questions as they appear in the control panel. So if you have a question, please don't be shy and type it in the control panel. Any question is a good question. The other attendees will not be able to see your question, so it is sent directly to Dr. Bothe and me. If you cannot see the control panel, just single-click on the single arrow on the top of your screen. In case your question is not answered at the end of today's session, Dr. Bothe will respond to you within the next 24 hours via email. If you're experiencing technical difficulties, please send the host a chat message, and our staff will be able to assist you. So let's get started with the webinar. We hope you will enjoy this webinar about the significance of genetic background of mouse models. Dr. Gerald Bothe holds a PhD in animal physiology and spent his postdoctoral fellowship at New York University, where he developed gene-targeted recombinase expressing mouse lines. He then worked at Lexicon Genetics in the field of gene targeting. He has been working at Taconic for over 10 years now, in the areas of mouse behavior, genetics, and the development of PCR assays for gene mutations and mouse pathogens. In this webinar today, Dr. Bothe will talk about the significance of genetic background and microbial status of mouse strains for the interpretation of experiments with laboratory mice, and how to eliminate or control genetic and microbial variation. For example, the tools for the analysis of the genetic background of mice have improved significantly over the last few years and now include a variety of SNP panels which are useful for speedy backcrossing or QTL analysis. And with this introduction, I would like to hand over the word to Dr. Gerald Bothe. Please, Dr. Bothe.
Hello, good morning everybody, or whatever time it is in your time zone. And what I wanted to talk about today is about mouse identity, basically, with the title "A Mouse is Not a Mouse is Not a Mouse," which is, of course, a variation on that famous quotation by Gertrude Stein, which said, "A rose is a rose." And so first I wanted to mention all the people who helped developing the concepts and the technologies I'm talking about here. And this is among the Taconic Genetics Group, it's Anna Perez and Paige Hinchy. Among Taconic R&D, it's Jan Gray and Michael Young. Taconic Molecular Analysis Group is Kim Mullinex, Rebecca Farinacci, Jamie Rusconi, and Steve Feston. And so what is it that determines the identity of a mouse? So, and how is it important in life science? So genetic variation is a basis of any experiment that you do in biology, because the genes determine the phenotype, and this is either the question you are addressing in your experiment, or it's a confounding factor if one gene interacts with another gene in an unexpected manner. Therefore, I say a mouse is not a mouse is not a mouse, because the genes that mice have are different depending on their origin, where they come from, and it's not only the skin color or the coat color that is important in the mice, but all the genes that they have, all 25,000 of them. And then there is an unexpected additional factor, and that is that mice have bacteria, and bacteria have genes. So actually, for a mouse to be healthy, The mouse doesn't need only the 24,000 genes or 25,000 genes that it has as of its own mouse genes, but also about a similar number at least of genes by bacteria that live in the gut and that help digesting the food. In addition, of course, a mouse can have millions of additional genes if it has a variant microbiota. So here are a few mice, and they all look the same, but they are all very different. So on top we have a wild mouse, unknown, that has an unknown genetic background, lots of mutations, has heterozygosity in its genome, it has bacteria, it also has viruses. Then comes a mouse of the inbred strain CBA, so this one is all homozygous in 99.999% of the loci. But it's different from the similarly looking 129S6. So the top 129S6 that has many bacteria. Then we have a 129S6 that has a restricted bacterial flora, so there has something been done to control the bacteria variation. And then the last one, unfortunately, it's 129S1, so that is yet another strain, somewhere in an animal facility in this country, where that has a virus, because the PI's child has a hamster, and the hamster have lots of viruses that also infect mice. So the PI carried the virus from the apartment to the animal facility in his mucous membranes and infected a mouse. That can happen. So if we go back to the natural population of mice, these are messy. They are just as human populations are messy in terms of their genetic makeup and also in terms of their pathogens and bacteria. They have varied, undefined symbionts and pathogens. They have genetically subpopulations and subpopulations. In mice, even more so than in humans, because mice have real subspecies. They have selective pressure, so that the genetic makeup of a certain population changes in a targeted manner. They have random drift. They have migration of mice from one population to another, bringing the bacteria, pathogens, and the genes with them. They have new mutations, of course. And as they have a lot of heterozygosity in their genome, we find sometimes overdominance. That means that a heterozygote is fitter than either homozygote. The heterozygotes are abundant, and fitness varies according to circumstances. So that means you have different conditions in your lab, in that lab that nature is. If it has cold winters, then different mice survive well than in warm winters, for example. So this is life, and human populations are just like it. But if we want to do research on mice, and want to have that research to have any meaning, then we need a way to standardize those models and either eliminate or control variation. So the steps to good experiments with animals are therefore that number one, we have to control the pathogens. Number two, we have to control the symbionts, the symbiotic bacteria in the gut and elsewhere. We have to eliminate or control genetic variation of the experimental animal itself. And that control of genetic variation comes in many different flavors. So you can control genetic variation by adding or taking out a gene. That would be transgenics and knockouts. You could move an interesting mutation from one mouse line to the other, changing the genetic background. You could do hybrid strains, meaning combining interesting genes from two populations and also increasing the heterozygosity of those strains. You could do mutagenesis in using new mutations. And of course, all this has to be controlled by genetic testing, or often also phenotypical testing, to make it a controlled experiment. So now the step one of this is to control pathogens. Taconic developed first that concept of a barrier unit that was in the 1980s. So all animals are enclosed in a secure unit that has filtered air, and where all supplies are autoclaved or irradiated to exclude pathogens. So humans accessing the facility have to shower and change clothes, or are completely kept out of the small units as glove boxes, which are also known as isolators or biobubbles. People from dirty environments have to take a cleanup time out of 5 days before they can enter a facility. No rodent pets are allowed for employees. That is indeed a ground for dismissals in most of the big animal suppliers, and also at Taconic. And for higher health status in animals, so if you want even to go beyond that and keep even mildly disadvantageous bacteria out, then one has to make technicians use spacesuit-like Dryden suits that filter the exhaust air. Then one has to control the health status, to introduce a system of tests and definitions that make sure that pathogens are excluded. And also one has to apply notobiotics, meaning germ-free rearing of animals. So the barrier concept is shown here in an aerial picture of the largest Taconic production site in Germantown, New York. What we see here is an administrative building, and then there's lots of barn-like buildings around. It really looks like a farm, and each one of these buildings is a barrier unit. The larger buildings, like here and here, have biobubbles, isolators, or a central autoclaving supply and office facilities. Any employee can work only in one barrier on any given day, and all supplies are autoclaved or irradiated. So that's the way to control the pathogens. Then the testing to make sure that this was successful is health testing. So we do periodic health tests at 2, 6, 13, 26, and 53 weeks intervals that form the International Health Monitoring System, or IHMS. And IHMS exceeds the European PHELASA guidelines, for example. And common pathogens are tested every 2 weeks, and the rare ones in longer intervals up to 1 year. So should there be a pathogen break in a facility, it is eliminated via a test and cull procedure. We had no virus break, though, for 30 years, but bacteria have occurred infrequently in the past. In that case, customers are informed immediately by email what has happened and what we are doing to address that problem. So the reason why viruses are easier to control in many aspects than bacteria is that mouse viruses usually don't multiply in humans, and therefore humans are no vectors for mouse viruses. That means if you in your facility or we in our facility make sure that there is a barrier around, that the air is filtered, and that no wild mice are entering and nobody has rodent pets, then it is relatively easier to control for viruses. Contrast this with bacteria. There are many bacteria that are opportunistic and that will grow in the soil, but also on the human skin and in mice, and will attack immunocompromised mice, and therefore it's more difficult to keep bacteria out. Completely keeping bacteria out needs the special procedure that are coming somewhere in the next slides. So here's another picture of doing microbial testing, much of which we are doing in the classical Petri dish style, because there you can detect also what you are not searching for. Just any growing bacteria are detected on Petri dishes. So now what this until now didn't address is how do I get clean starting bacteria? Of course, this system works only if an initial supply of clean animals is available. Else, if you put dirty animals in a clean facility, then they keep their pathogens and bacteria, and the facility becomes dirty. So, therefore, what we do is that we embryo rederive all mice before they are allowed to enter a taconic barrier. That means mouse embryos are produced in one female, then they are flushed out of the uterus, transplanted into a germ-free recipient animal. The rats are still caesarean re-derived, although there is lots of work going on to make this all embryo re-derivation as well. And we verify the absence of all known pathogens by testing after this re-derivation, and the initial result then is germ-free animals. We have a separate re-derivation facility, so all around here is empty space, and the road leading to this facility leads only to this facility. If you want to get to the main barriers, the main animal facility, you have to drive about a mile. Now, how do we handle the germ-free mice that come out of the re-derivation step? Well, they are handled in what is called a biobubble. So this is a plastic bubble where HEPA-filtered air is pumped in, and where you don't have direct access to, but it's a glove box type setup. And this can keep mice sterile. Now, we need a step number 2, and that is control symbiotic bacteria. This is needed for a number of reasons. Number 1 is germ-free animals are not healthy. They have intestinal problems. They don't completely digest food. Some vitamins are in short supply as they are not provided by intestinal flora. In mice, they have an enlarged cecum that often results in a cecal volvulus, or so-called intestinal strangulation, where they get a blockage of the intestines and get up to 15% death rate. So therefore, animals that leave, and also animals that leave the germ-free environment, are extremely susceptible to infection. For example, LD50 in Salmonella is reduced 100,000-fold due to the loss of colonization resistance. In normal animals, the existing intestinal flora tries to push out all alien invaders that are pathogens. Also, germ-free mice have an understimulated immune system. The solution to all this is that you add carefully selected symbionts to germ-free mice. And the cocktail of symbionts we are using is called Alder-Schädler flora, which is a mixture of 8 symbiotic bacteria that we add after re-derivation. These species were selected to fill important ecological niches in the intestinal habitat, but also to be recognizable by microscopy and culture, so that you can relatively easily say, do we have the right bacteria or not? All these species grow anaerobically, like the majority of gut bacteria do. Only 2 of them can also grow in air, so that's the group microaerophiles. We have 2 normal anaerobes in there, plus 4 extremely oxygen sensitive anaerobes. These are spore formers that keel over when they have vegetative cells. These vegetative cells die immediately if they are exposed to air, and only the spores are resistant. So then, if you want to keep the flora defined as having only Alder-Schädler flora, then you still need to keep the animal in a biobubble or isolator, because else it will soon acquire other symbiotic bacteria. So therefore we have several levels of animals that are varying in price and in the effort that is done to exclude bacteria. In defined flora animals, they are still kept in isolators. We allow no other bacteria. In restricted flora animals, we allow the opportunist Klebsiella. We do not allow the opportunist Klebsiella, Oxytoca, Pneumoniae, Pseudomonas aeruginosa, and Staphylococcus aureus. And also viruses and all other pathogens are excluded as well. In murine pathogen-free animals, we exclude the viruses and pathogens, but not the opportunistic bacteria. Of course, therefore, theoretically, the defined flora animals would be best for everything, but they are so expensive that it's not practical to use. And the restricted flora animals are then the next best things, but murine pathogen-free animals can be used in all experiments that do not involve immunocompromised mice, because the difference between symbionts and the opportunistic pathogens like Klebsiella, that becomes only visible if the immune system of the mouse is compromised. So now finally we arrive at the genetic variation of the mice themselves. So after pathogens have been eliminated and variation in symbionts has been dealt with, we can turn to the animal itself. Many questions can be investigated using genetically homogeneous lab animals, and I will get to how to produce and keep them later. And these strains are inbred and related strains, which are produced by inbreeding brother to sister over 20 generations or more. We have a wide variety of inbred strains, such as 129 and B6 and BALB-C and so on. So there are some other models that are not completely genetically homogeneous, but that have controlled, stabilized genetic variation. These are needed for some experiments, and I will get back to those at the very end of this talk. But first about inbred strains. So as I said, we produce them by brother-sister mating over at least 20 generations. All traditional inbred strains are derived from pet mice, which are also called fancy mice, of the early 1900s. After 20 generations of this inbreeding process, 98.7% of all loci are homogeneous, or 1.3% of original heterozygosity remains. After 80 generations, we have 3.9 times 10 to the minus 8 of original heterozygosity remaining, which is practically nothing, because mice have a few million positions in the genome in total where they vary from each other. And of course, 1 million times 10 to the minus 8 is practically nothing. That means that those inbred strains have no original genetic variability remaining. But of course, there's always new mutations. So each animal will carry approximately 60 new mutations, and of these will 15 become fixed or homozygous in the germline. And at any given time point, there will be around 120 loci or so that are heterozygous due to recent mutations. This all depends on the mutation rate, and the details of the mutation rates, also the different mutation rates that are there in mice, are still being discussed. So these numbers refer only to SNPs, to single nucleotide polymorphisms, and may change in the future. Don't nail anybody on the exact numbers like that. The major inbred strains are C57 Black 6, also short known as B6. This is, so to say, the standard model. And the standard genome in the database is a C57BL6 model. About 150 generations ago, the B6 line split up into two major substrains, and this is B6J, C57BL/6J, and B6N, for example, our B6N tag. These substrains have some differences. For example, the BL6J, but not the BL6J bomb tag that we have, carries the NNT deletion mutation, which changes the response to high-fat diet and glucose metabolism. Then there is a group of strains that is called 129, and they have today 129 and a letter and a number, for example, 129P1 or 129S6 SVEV tag, that is Taconic's line. And here I can short say what is the strain definitions. And so this here is actually the strain name. And then down here you have where it has been bred. Now if the same lab has produced more than one strain from one pair of animal or from one small stock of animals, and that comes first after the slash and then the name of the keeping lab, which is here Jackson Lab. So for example, in the C57BL, there's also a C57BL10, not only a C57BL6. So here is a BELP-C, which is another very old line, and then there is DBA2. That's another one. Dilute brown is the meaning of this, and there were two strains produced from this stock. One is DBA1 and one is DBA2, but DBA2 is used more often. Each strain carries a unique combination of gene, and that's what we call the genetic background. So the genetic background is the same for every mouse of this inbred strain. Generally, we talk of a strain if it's inbred, and if it's outbred, and therefore genetically heterogeneous, we call it a stock. And the genetic background influences the phenotype of any transgene that is produced using this gene. Some transgenes are lethal on a certain genetic background, whereas the animals are viable on other genetic backgrounds. And one example for this is the Swedish mutation of the amyloid precursor protein. Amyloid precursor protein, or short APP, is a very famous gene because it is most probably the causative agent for Alzheimer's disease. And so therefore, transgene was made from a mutation of that APP gene that produces familial Alzheimer's disease. And that's a Swedish mutation. So after these mice had been produced on a mixed genetic background that consisted of B6 and SJL, people tried to move the mutation to a pure B6 background. But when they did that, more and more of the mice died, and it turned out that the gene is lethal on a B6 background, whereas it's viable on a mixed B6SJL background, and it's also viable on other backgrounds. In mice with a mixed B6SJL background, the mortality of transgene carriers is not higher than that of non-carriers, and therefore, currently, this transgene is either propagated on a mixed B6SJL background. We do that by mating B6SJLF1 in each generation with a transgene carrier, so that in the end this genetic background resembles a B6SJLF2. And we also have a line where the transgene is propagated on a 129S6 background. So now, as we know, we have a number of inbred mouse strains that all are different, react different to environments and to experiments. We need a way to tell strains apart. In the old days, that was done by biochemical markers, isoenzymes, or microsatellite markers. For isoenzyme analysis, you need to, for example, dissect an animal, take out the kidneys, and then run a protein gel and look for enzyme isotypes on that gel, which is, of course, a very involved procedure, and you also have to kill an animal for that. So microsatellite markers are better because those are genetic markers, so you can type them from a tail clip or from blood. But today, the best way to identify strains is through single nucleotide polymorphisms. One example of that is the sequence below. So you have flanking sequence, 5' and 3' of a change in the genome. And this change is one base is replaced by another one. So for example, a G is replaced by a T. This can be disruptive to the phenotype or not, but in most cases it's not disruptive to the phenotype because this change is just somewhere in the genome where it doesn't matter that much. So you don't have any overt phenotype for, say, 99.99% of all the SNPs that are known. But one can use them to characterize the genetic background of a mouse. So when we have a number of genetic marker panels, so generally to tell strains and genetic background apart or localize mutations, at least around 90 SNPs or so are needed. And we use 96 for practical purposes because of the 96 well plate. Therefore, we have a 96 marker panel that we use for general genetic monitoring. But this one is not suitable for following backcrossing or localized genes because we have too low resolution in that. And also, as the markers were chosen to tell strains apart, the markers we used are unevenly distributed. Because, say there's a group of markers that tells all the 129 strains apart, but they are clustered on one chromosome. Then we have them there, and we have an empty region on another chromosome. So this one is for genetic monitoring, but for nothing else. Then we have a low resolution mapping panel, which is 377 SNPs, that can be used for accelerated backcrossing and low resolution mapping. But the best panel for that purpose is a mid-resolution 1449 SNP panel for backcrossing and mapping of Mendelian mutations, and also for the identification of quantitative trait loci. The technology that we use is shown in this picture. So we start with genomic DNA which is put on a solid support just to make it more easy to handle this. We then have 3 primers, 1 primer that is common to each locus, 1 primer that is specific to one of the mutations, and 1 primer that is specific to the other mutation. In this case, the substrate, the genomic DNA, has an A here, so the primer, mutation primer with a T at the end binds. Then the primer is extended just to where the other primer sits, and then it's ligated here, so that we get this here as a first product. Then we do PCR with universal primers and get this whole thing here with the green fluorescent label as an end product of a PCR reaction. At this stage, we got rid of the beads in the wash step and work in solution. So then we hybridize those markers to a second set of beads, and this has label sequences on the beads and a complementary label sequence that is tailed to the locus-specific primer is here, so that we now know bead number 1101 is carrying here, for example, only allele B, or in this case it's carrying allele A, or here it's heterozygous. So with these beads that have the label or address sequence on them, we can identify each mutation. And for one mutation, RS327074440, we then get a picture like this. So now for this SNP, each point is a sample, and there are 1,148 other pictures like this in this data set. So let's say this sample here is a homozygote for the blue allele, for allele B. This sample here is a homozygote for the red allele, which is A, and here are heterozygotes. And the graph is just norm intensity A versus norm intensity B. The coverage of the 2 larger of these panels that can be used for linkage mapping and for QTL analysis is shown here. And one can see that the high density panel is definitely better than the low density panel. The low density panel still has some small gaps in there. So the higher density panel was then looked at and used for analyzing a number of inbred strains that are commonly used in the mouse field. And we have here each strain compared with another strain, and what is in the table is the number of differences in this 1449 SNP panel. And if there is a lot of differences, it's blue, and the other colors have less and less differences, with red being the lowest differences. One can see that the number of differences in the SNP panel roughly follows the relatedness of those strains. For example, here's that group of strains that is commonly used to make knockout mice, 129P1, 129S6, 129X1, and they have relatively few mutations distinguishing each pair. Then we see here AJ and BALB-C are fairly closely related, therefore fairly few differences. Also, what has few differences is CB57BL10 and BL6, and that's no wonder, because as I said earlier, these come from one small population of mice that was used to make both of these inbred strains. The same is true for DBA. DBA1 and DBA2 also have very few mutations differentiating them, because they come both from the same small population. So, and then 382, here's another example where there's relatively few differences. But in all other pairs, we can, in all the blue pairs, we can confidently use this for mapping mutations and for doing QTL analysis. So the first application I wanted to talk about is backcrossing. So backcrossing is used if you want to have a mutation on a different genetic background. For example, if you have the APP Swedish mutation on that mixed B6SJL background, and you say I want to move it to a homogeneous genetic background, then you cannot move it to B6 because it's lethal on that background. Let's say we want to move it on 129 strain, then you have to backcross it. To backcross it, you made a carrier of that gene to a mouse of the desired background, so in that case an APP Swedish mutation carrier to a 129 mouse, and then you repeat that process several times. If you do that 10 times, the strain is called a congenic. So here is that in a diagram. One sees here's the donor that has the interesting mutation, and the recipient is the strain where we want to move the mutation to. So in the F1 it's 50-50, and this F1 animal is mated to another recipient. Then the N2 already has only a quarter of all the genetic material from the donor, or equivalently only 1/2 of all loci are heterozygous. Made that again and repeat and repeat and repeat until it's a congenic. So this step of a quarter, an eighth, a 1/16th, and so on of the genetic material, that applies only to loci that are not linked to the mutation of interest. The loci that are on the same chromosome as the mutation of interest and are close to the mutation of interest are linked, and they are eliminated much more slowly. They are eliminated only if there is a crossover close to the transgene or the other mutation of interest that you have. So therefore, in a congenic strain, it usually looks like this. So you start with a donor that has the gene of interest and what you could call a passenger locus close by. If you backcross this to the recipient for getting an N10 mouse, this N10 mouse has very little genetic material outside the region where the mutation of interest is, but there are about 20 centimorgans or 1.3% of the whole genome around your interesting mutation, because this is linked genetically. And these loci here that travel together with the mutation of interest, those are the passenger loci. So now the production of congenic strains is a time-consuming process. It's about 2.5 years to N10. In each generation, though, there is a random distribution of offspring. Some offspring have more donor genes than the others. So therefore, if you have a high resolution or medium resolution SNP panel, you can use that panel to select the best offspring and accelerate that backcrossing. You can also use the same SNP markers to reduce the size of the donor segment, which improves the quality of the congenic line by eliminating many of those passenger loci that I showed in the last slide. We achieve optimal results with about 20 males carriers tested per generation, but you can reduce this to about 5 if cost is a problem. Only the overall quality, say, reduction of the donor segment is proportional to the number of carriers that you test. So and then in a speed congenic strain that you produced with this method, you get to the equivalent of N10 in half the generations. It means that congenic is actually an N5, but it's equivalent to N10 or better. And that goes the following way. It has slightly more material outside the mutation of interest, maybe, say 0.3% instead of 0.2%, but it has a smaller donor segment. And that more than makes up for any small difference that you might have in the non-linked loci. So another application of SNP panels is genetic markers used to map gene or to analyze QTLs. So mapping is generally used for finding mutations on the chromosome. Say I know I have a phenotypic mutation, I want to know on which chromosome is it and where on that chromosome, then I use the mapping process to find that out. If you have multiple genes that each only have a small influence on phenotype, you do what is called a quantitative trait loci analysis. The approach is always to create a cross between a mouse line that carries the gene and the phenotype, i.e. gene and genetic background together produce the phenotype. Then you make an F1 and look, is it uniform, or is it that only males show the phenotype? Does it matter whether the mother of the F1 is strain 1 or strain 2? What's the distribution of the phenotype, meaning do I have 100% of the animals carrying the phenotype, all or nothing? Or is in the F1, is there only 90 of 100 animals carry the phenotype and the other ones not? That would be incompletely penetrant and other questions like that. So then you produce an F2 by mating 2 F1 animals and do again, how is the phenotype distributed, what measures the phenotype and determines SNP marker status for at least 100 individuals. And then one can put that into a program that performs a QTL analysis. Such programs are available on the web. And the result is the position of the gene of interest and of modifier loci if they exist. Of course, if you are sure that it's only one mutation that influences your phenotype, you can also get away with less than 100 animals. So, and here, right at the end, I wanted to get to another type of models that are more rarely used for actual experiments, and that is outbred stocks. So there are some questions that cannot be studied in inbred strains, and this is all those that need genetic diversity in starting populations. Remember, in inbred strains, the genetic diversity is excluded. Example for that is if you want to try to select for an alcohol-preferring phenotype by, in each generation, selecting those animals that prefer alcohol against water. Then you have to have a genetically diverse population to make that experiment meaningful. And at the end, you will have a population where the genes that drive towards alcoholism are enriched. So, yeah, you start with a genetically diverse population of mice. And then you have to maintain genetic diversity by rotational breeding of hundreds of animals. And you have to avoid genetic drift by a large population size. And no selection until your selection experiment starts. And then you have to keep the population large enough so that the selection experiment only selects for the genes that are related to alcoholism, for example, and not other genes randomly. So the resulting population will maintain diversity and multitude of haplotypes that is typical of natural populations. On the other hand, they avoid the chaos that is connected to natural populations because you don't have random migration from one animal facility to the other and things like this. The use of those outbred stocks is toxicology and other drug research, and as I said before, the selection of interesting phenotypes. Then outbred mice are also used where you only need breeding performance. For example, foster mothers for inbred mice during re-derivation. These are outbred mice that are just used for their gentle behavior and their good breeding performance. So, yeah, I wanted again to thank everybody who worked with me on all these questions, and I wanted to open the thing for discussion.
Okay, thank you, Gerald, for the interesting presentation. I think we'll immediately go over now to the questions and answer session. So a question is here. How many SNPs would have to be tested to confirm the identity of mice?
Yeah, so that is an interesting question because you can always ask how closely is the most closely related strain that you want to differentiate from. For example, if you know that it can only be either a 129 mouse pure or a B6 mouse pure, then you would only need 1 or 2 SNPs to do that. But if you have a situation where there are multiple strains in your animal facility, then it's already advisable to take a large number, for example, the 96 SNPs that we have in our panel. Now, if you find anything suspicious with such a panel, for example, that a B6 NTAC mouse is in reality half or something like half B6J, so then you get to the question, are there 2 substrains that are mixed up? And as substrains are really closely related, then you would need to use another panel to really solve that question. Also, you need much more markers than this generally genetic monitoring panel of 96 if you not only want to know whether there is genetic contamination, but if you know exactly how many % genetic contamination is there, because then you get into statistical questions. And so for all normal genetic monitoring, I would take something like a 90 or 96 marker panel, and if there's anything involved like how many % contamination do I have, or there may have been a mouse entering that population 25 generations ago, then I would take the large panel to solve questions like this. Because for example, in the case of exactly how much genetic mixing is there, then you have a quantitative question. You need a bigger precision. And if you have a mouse entering the facility 25 generations ago, then you have kind of a dilution problem too in some cases.
Okay, thank you. There's another question. So do you need to use males only from F1 in the congenic process?
Actually, if you want to make the or the whole genome congenic, then you have to have in that whole process one female as well. So for example, if you make the first generation, the F1, you would have a female donor to produce the F1, then the resulting mouse, and if you have the other way around, right? If you have say you backcross B6 to 129, then in the first generation you produce the F1 with the donor B6 and the recipient 129. So the donor would be a male and the 129 is a female. Then in the F1, the Y chromosome is still from the donor, and the X chromosome is already from the recipient strain. So that means that there is no crossover between the two. Then you do the backcrossing the whole way through to N10, and then the best is to use a female offspring at one point and a male from a recipient strain in the last backcross to get rid of the Y chromosome that is originally still from the donor. So for most of the backcrossing, you use males for practical purposes because they produce much more offspring. But then in one generation, you use a female to finally get rid of the Y chromosome.
Okay. Another question is, that's an interesting one. Are there any transgene strain pairs where the actual mechanism of lethality related to that particular background is known? Has anybody ever found that out?
Yeah, I don't know of any good example for that. One would have to ask around for that, whether it's really completely known where the lethality is caused by. Yeah.
Maybe there aren't actually very many examples, but yeah, if we find some, yeah, we forward to the question. Yeah, we'll get back to that if we find some literature about that. Another question also with regard to the congenics is, when you have a knockout mouse line, do you actually have to make congenic strains? Couldn't you just make a cross with B6 and use the wild-type litter mates as a control?
Yeah, so this one came up in some earlier talks, so I even have a slide for it.
Good.
Slide for that. And
these things are better done by graphics, actually. Yeah.
So, and here's what's happening there. If you have a knockout made on 129 line, you cross this to a B6, then you get an F1. Now in the F1, of course, everything is heterozygous. So that's nice. Heterozygous, one strain and the other is also a defined genetic background. Actually, you can use that for some questions as well. You can use F1 hybrids for many experiments because they are also completely genetically defined. But then in the F2, something nasty happens, and that is if you cross those two, get an F2, then the surrounding of the gene of interest, which in this case is a knockout, is still from the original 129 knockout strain. That means that the F2 knockout carrier is not only homozygous for the knockout, but also homozygous for all of those passenger loci. And that can have a negative influence on your experiment because the so-called control, the wild type, is B6 on all those loci.
Okay.
The reason why it is better to use congenic strains. Of course, if the effect of the knockout is very large, then it doesn't matter. For example, if you knock out a gene that is important for the formation of heart muscle, then all these will be embryonic lethal anyway, no matter what's the passenger loci, and these will all be viable, so that's not a problem. But if you have some delicate behavioral patterns and say there is a locus for sluggish movement somewhere close to the knockout, then you can confound your experimental data.
Okay. Maybe we take one more question. So here's another question. Should all researchers studying the phenotype effects of their knockout animals do these SNP assays to eliminate any possible background effect to such observed phenotypes?
Yeah, so I'd say one should at least do a background check on mice when you receive them from another lab or after you have bred them for a few generations and so on. You can also do it like this. If you say breed up a population for experiments, you can take the breeders. You can check on the breeders, and then that means you do two test panels, and then you test, and then you can, from then on, you can use the animals in your experiment, the offspring in your experiment. So yes, there should be a background check on all your animals before they enter the experiment, but there are ways, there are strategies of breeding and of testing that will reduce your cost a little bit.
Okay, I think our time has gone. We went a bit over time, but I think we'll stop here. So any other questions will be answered after the webinar by Gerald. He will be happy to do that. So for this, I would first of all thank you, Gerald, for the interesting presentation. Thank everybody for your interest, your attendance. We hope you enjoyed the webinar. We hope you learned something or got some inspiration for your work. Also, please let us know by filling out the questionnaire after the webinar. And yeah, we'll hope to welcome you again at another webinar. So please have a look on our webpage. Hang on and hope to welcome you again. Bye-bye.